Mr. Chairman, ladies and gentlemen, in addressing an audience of chemists, I must remind you that for us in the medical school, chemistry is an applied science. We are obliged to work with a test tube in the one hand and a frog, a mouse, a rat, or a rabbit in the other. Our field of work lies largely in the borderland between physics and chemistry and biological and medical science. I can only select a few examples from among many researches that have been carried on in the last 40 years by way of illustration of my statement of our great dependence in the field of biological research on chemical and physical science. One of the topics that I should like to present to you concerns what is called vividiffusion. I should like now to take up with you another topic, also of great interest to chemists. This refers to the subject of animal poisons. You are all aware that chemical warfare was invented long ago by the lower animals, that many poisonous substances are produced by them and are utilized either for offense or defense. Let us consider for a moment the ordinary toad. It is known, has long been known, to laymen as well as to physicians, that toad skins contain poisons. For many centuries, extracts of toad skin were used by physicians in combating the dropsy associated with heart disease. It was the regular practice of physicians in Europe and elsewhere until about the time of our Revolutionary War to make use of toad skin infusion or extract in treating cases of heart disease. Now, I have here a picture of a large toad that is found in South America and in the tropical islands of the Caribbean and in Mexico, a toad called Bufo agua. It is a veritable giant of its species. This toad secretes from little glands in its skin, which are especially numerous in the neck region, and are called, therefore, parotoid glands. I say this toad secretes a substance which looks like a cream, a heavy cream. The savages of the upper Amazon have long used this secretion as an arrow poison. They obtain it in this way. They take one of these large toads and shove him up into a hollowed out piece of bamboo and then hold him over a warm fire until he becomes so uncomfortable that the creamy secretion drops from his skin. These savages then dip their arrowheads into this creamy secretion and let it dry on them. It dries like a firm varnish, and it is, of course, very deadly either to their enemies or to large animals that they hunt. It had been quite forgotten in the past century that toad poisons have a valuable remedy in their, the toad poisons contain a valuable remedy. I was able to collect from samples of bufo agua sufficient amount of this creamy excretion in order to analyze it chemically. And two substances were obtained in beautifully crystalline form. One, the substance that we shall hear about in a moment, which is also found in the suprarenal glands of all animals, including ourselves, a beautifully crystalline principle which is known in the pharmacopoeia as epinephrine, which goes by such names as adrenaline, adrenin, suprarenin, and so on. Well, in addition to this crystalline principle, I was also able to crystallize that particular constituent of the toad secretion which has been so much used in the past as a heart remedy. I found it to be a beautifully crystalline substance with a molecular formula C6H12O2. It's a singular thing that this crystalline principle, which is the heart remedy of all toad skins, is closely related, fundamentally related, I had better say, in its chemical constitution to the digitalis glucosides which have replaced it. It was only about in 1780 or thereabouts that the English physician Withering discovered the properties, the medicinal properties of digitalis. And since then, that has taken the place of the toad poisons. The suprarenal glands are small organs shaped like a cocked hat, each lying above its kidney, each lying above the kidney on its own side. This suprarenal gland is a double organ and elaborates in its medullary or central portion a well-known crystalline principle which I spoke of before as being one of the substances excreted by the large tropical toad, bufo agua, one of the two substances, too, that constituted that arrow poison that I spoke of. This substance is a simple aromatic amino alcohol, dihydroxy methylamino ethylol benzene. It was prepared by me many years ago in the form of a monobenzoyl derivative and was later isolated by Takamini and Aldrich in its native form without the benzoyl group. And later the chemists, Dakin, Stahl, Flecher, and others, cleared up its constitution and was synthesized by Stahl and Flecher. This is the substance, as I've already stated, is known as epinephrine or adrenaline. Another instance of these chemical substances concerns the pancreas and its hormone, insulin, or chemical messenger. I rather like that term. I rather prefer it to the term hormone. Those are as well put it. Nowadays, the dead in mind and the deformed in body may be restored by the magic wand of science. Now, insulin is only one of the constituents of the ordinary therapeutic extracts which physicians use. It is necessary. I said somewhere, one of my papers, a mandate is laid upon us to isolate, if possible, the actual effective hormone, to learn about its chemical structure. When that has once been done, advance is more rapid in many ways, at least along scientific lines of research. There is thrown upon the screen a picture of crystallized insulin. Some years ago, I obtained this substance in the form of beautiful crystals belonging to the rhombohedral system. You will see that when they're caught standing on end, you have 6 crossbars. That, of course, represents the projection on the median plane, when the photograph is made, of the 6 angular edges. Now, this crystalline substance is of protein nature, and it is the first hormone of the protein class to be isolated. The Chemists Association, who have been associated with me in this work, such as Dr. Jensen, Dr. Geiling, Dr. Wintersteiner, and others, have been engaged in determining the chemical constitution of this crystalline protein. I can only say here that we have advanced to a considerable extent and have split up the molecule so that we can account among the split products for at least 40% of the total nitrogen. And so far we have found such amino acids as cysteine, arginine, histidine, leucine, and tyrosine. But the substance still requires, this principle still requires much further research for its complete understanding.